Oh, good day, good day, good day. As I promised last time that I'm gonna try some questions from past papers, and then in case if some of you are surprised why I'm only doing these certain sections, I'll advise that you go through the playlist which is under genetics. So I'm doing sections on that order, and then these questions that I'm doing today will be based on the sections I would have covered by the time you will have seen other lessons on the playlist. So don't be surprised why we're not covering paper one, why we're not doing evolution or meiosis. I haven't done those sections yet. So I'm just going from that section of DNA, and then I'll cover other sections as time goes by. So without wasting any time, I'm going to go straight to the three questions. So first of all, they're giving us a DNA molecule. So they're telling us it's a DNA molecule. And we know DNA molecule has got two strands with double stranded. We also know that it's got thymine, which they're showing us here. So we know that if it's a thymine there, here we must have adenine. It should be A. So in full it's adenine, but it should be A. We've got double bonds there, triple bonds, it's fine. We've got a nucleotide, which is having the phosphate group, which is P. It's having a phosphate group, which is P. So let me zoom out. We've got a phosphate group, which is P. And then that's P there. We've got our sugar. And then we've got the bases. Sharp, covered. So first thing first, they're asking us, what is the natural shape of DNA molecule? We know that DNA, as it was shown by Watson and Crick, that DNA is double helix. Sharp. That's the first one. DNA is double helix. Sorry for the big graph there, but it's fine. So second point, sugar A. They're asking for sugar A. That's sugar A there. We know that sugar A is deoxyribose sugar. That's deoxyribose sugar. Sharp. Then next question, the base B. Base B already, as we showed before, is supposed to be adenine. And they're also asking us to write our answers in full. So don't just write the symbol A, write in full. Just adenine. Sharp. And then bond C, we know in DNA we've got the weak hydrogen bonds. And then in amino acids we've got the peptide bond. But in DNA we've got the weak hydrogen bonds. Hydrogen bonds. Sharp. And the 1 mark. So they're asking two visible reasons why the diagram is DNA. Remember, we've got visible T there. We've got thymine, which is visible. Thymine that is visible. And then also I've got the double stranded. There are two strands there, so it's also double stranded. Those are my 2 points or 2 reasons as they're asking. So 2 structures in a non-dividing human cell where DNA will be found. So you'll expect to find your DNA. Remember there are 3 places where you can find your DNA. You can find the nucleus, mitochondria, or chloroplast. So here they're talking about the human cell, and we know chloroplast is in plants. So in a human cell, it can be in a mitochondria or nucleus. So that's it. So that's a good 1, 2, 3, 4, 6, 8. That's good 8 marks. So without wasting any time, I want to go to the second question. Second question. Okay, they're giving us a crime scene. They're saying that in a crime scene there were 3 men who were murdered. Okay, one of them is suspected to be Anna's father, they could not be physically identified, and DNA fingerprinting was used to find Anna's father. Okay, fine. They're giving us DNA fingerprinting bars of the mother, Anna, and 3 men who were murdered. So first thing first, you must always do with DNA fingerprinting. For example, here you need to compare with the child. So we know that a child will get the bands from the mother and one of the father. So we're going to compare Anna's bands with her mother's and all 3 men. There must be one man who must either, if the mother doesn't have the band, that man must show other remaining bands. So let's check the first band. Mother is not there. So this band is coming from the father. This band is coming from the father because it's not there on her mother. So I go to each father. Number 1 is not there. It's not there in number 1. So fine. Number 1, I'm going to take you out since Anna has got the band. It's not there on the mother. It's not there on you. So it can't be you as the father. Number 2, okay, the band is there. 3, the band is there. Okay, 2 and 3 so far. There's a big possibility that 2 or 3 they are Anna's father. Then I go to the second band is there on the mother. Okay, fine. I won't go further since it's there on the mother. So maybe it's coming from the mother. It is not there on the fathers. That's fine. Then I go to another band. There is there on the mother. 2 and 3. 2 is there. 3 is not there, but it's there on the mother. So I can't also look too much into it. I want the bands which are not there on the mother, because they're the one that will tell me which father is giving Anna those bands. This band here, remember, number 1 is already out. I'm focusing on 2 and 3. This band here is there in number 3. Okay, so far this band is there on Anna and also is there on number 3. Also this one is there on Anna and also in number 3. Fine. The other band, this is the other band. So these 3 bands, 1, 2, and 3, they're not there on the mother. And all 3 of them are there on number 3. So it's clear now that number 3 is having all 3 bands which the mother is not having. So number 3 is the one who there's a big possibility that he is the father, because all the 3 bands which Anna is not having are there on him. So the first question, which one is more likely to be Anna's father? Number 3. Number 3 would be more likely Anna's father. Reasons, remember, first of all, Anna gets the bands from mother and the father. That should be a first reason. All 3 bands which are not on Anna's mother are there on man number 3. Second point. Lastly, because number 3 is having those bands which are missing, therefore we can conclude that he is more likely to be Anna's father. Those are my 3 reasons. And then give a reason why the evidence from DNA is considered reliable. Remember, DNA is unique on everyone. Every human being has a unique DNA, except for twins. Twins are the one who can have similar DNA, but everyone else, like me, I don't have a twin, so my DNA is unique. So because DNA is unique, they can use it for evidence because it's unique to everyone. 2 uses of DNA profiling. Remember, it can be used to identify genetic disorders. It can be used to cure genetic disorders. It can be used to find relatives. Here also can be used for crime scene. So they just want 2 reasons. So you can just list any 2, but if you list 3, always remember they mark the first 2. So your first 2 must be on point, because if you're not sure about your first 2, they're gonna mark the first 2. Unless you find someone who's nice, but mostly they mark the first 2. So that's 7 marks. That's 7 marks this question. So the last one that I'm going to focus on is protein synthesis. So we're going to focus on protein synthesis. So remember, it's transcription. We know this is in the nucleus. I'm assuming this big box is a nucleus. Maybe there's some nuclear pore somewhere there, since the whole thing is transcription. And we know during transcription, DNA goes to mRNA. And then is it DNA? Oh yes. You see, there's T there. There are a lot of T's there. So I know it's a DNA because there are a lot of T's. And this is the first strand here on top. I'm moving my first strand. And remember, they complement each other. Here it's A, here is T, G, C. Therefore, this is my second strand. It's DNA alone. That's why there's no U. It's only DNA. But then here I'm even use or uracils here, which means this is my RNA. So I know Y is RNA. This is my RNA. I'll just put an R to show that it's RNA. Here I just put D to show that it's DNA. Yes, that was terrible, but yeah. So the first question, identify molecule X. X is DNA. That's your DNA there. Give a reason for your answer. Visible reason. Okay, I can clearly see this T there, this thymine there, which means it must be DNA. Also, there's another strand. There are two strands also. That can be my second reason. In case they would ask for two reasons, there's also two strands. Therefore, DNA we know is double stranded. So there are two strands. And also this thymine. I think thymine is the safest one that you can go for. How many amino acids? Okay. Remember, each triplet makes one amino acid, right? Each triplet base will make one amino acid. Because tRNA will come and match with this mRNA. This is our mRNA in Y. So each tRNA will come and match with this mRNA. So each will form one amino acid. So I'm going to have one amino acid, 2, 3, 4, 5. So I'm going to have 5 amino acids. So it will be lovely, 5 amino acids. Okay. They're giving us in 2.1.4, they're giving us a table which is having codons and different amino acids. Okay, fine. So with reference to 2.1, which is this diagram, they're telling us that now we are forming CAC due to maybe it's a mutation or there's a change in the sequence. It could be mutation. And then now it's forming, instead of having TAC, we're having CAC. So now we know our DNA molecule, it's CAC. So we've got DNA as CAC. That's our DNA now. It's CAC. Let's put a D so that that's our DNA. And then we know it will go to mRNA. So mRNA, we know it will complement my DNA. So it will be G, U, and then G. So GUG, that's my mRNA. We know that it will go to tRNA, which will be C, and then A, and then C. So that's my tRNA. So basically now we're going to form this tRNA. So when you explain here, you must go the way I did. DNA, the mRNA that will form will be GUG, tRNA, CAC. So now I'm forming codon of GUG. I go to my table which is having codons. Where's GUG? There we are. GUG, it's this one. That's my GUG, which is my codon. It's valine. It's valine. So now I'm gonna form valine. So you must write that down that you're gonna form valine as your amino acid. And then if I'm having TAC, for example, TAC, I know my mRNA will be AUG. My mRNA will be AUG. I go and check, is it AUG? Oh yes, that's it. Methionine. So in a normal TAC, you form methionine amino acid, but then now because of a change, we're forming valine. So when you answer the question, you must give this order from DNA, mRNA to tRNA, and then tell them which amino acid will now be formed instead of forming the methionine. Sharp. That will be your 4 marks. So last question, write down the DNA that codes for alanine. So I go and check alanine in the table. Where are you? That's it. That's alanine. It's GCU as my DNA. So we know the codon will be CGA. CGA. So that will be my codon. CGA. That's it, guys. That's another 8 marks. And then also thank you guys for your participation. And if you've got any questions, feel free to ask on the comment section, or any other past paper that you have that you might want us to try. So feel free to send it on the comment page or section.